So today I'm going to talk to you about the DevoWorm 2025 update. This is going to be presented at the OpenWorm Foundation annual meeting, but I'm going to talk about it in this video. So one of our main activities for the year was to participate in Google Summer of Code, or GSOC. And so of course we've been participating in GSOC since 2017. And this year we had two scholars, Laylath Bharadwaj Baru, and his work was on modeling neural developmental programs, or NDPs, for C. elegans using growing hypergraph neural networks, or HNNs. So Paki Banchalia last year worked on neural developmental programs, forking that repository, and this year Laylath incorporated that work into hypergraph neural network models. And that was something that we've been working with hypergraph neural network models for a couple of years now. So we're getting that pipeline fortified with a lot of different methods and techniques. Jayadratha Gayan is the other scholar for this year. His project was DevoTG, and that's Developmental Temporal Graph Networks. So he was interested in building graphs from a secondary data set that sort of compounded over time, and we'll see examples of that later. So this is Laylath's project HNN NDP. This is using data from the interactive C. elegans lineage tree. This is the data set that is provided by Nikhil Bhatla, and he has an interactive lineage tree GUI that you can explore on the web. And this is kind of taking those data and building these kind of hypergraphs. So the idea is that you have these hypernodes, which contain a bunch of nodes, and then these edges, which are hyperedges, which contain a bunch of edges in them. And the idea is that hypergraphs are sort of like categories or groups of nodes, and so you can group them in different ways, and the individual nodes are connected to other nodes. So you have these hyper edges, which is like a bundle of edges that go from hyper node to hypernode. In any case, you can do a lot of interesting things with hypergraphs. And one of those things is analyze the lineage tree in new ways so that we can find relationships, spatial relationships perhaps, for different cells in C. elegans development. So this is what he calls a lineage graph. This is over time, so we have 3 time points represented here. We have the hypernodes colored in blue or green or red. And so each of these cells belongs to a hypernode. And then we have the edges between the nodes that are bundled into these hyper edges. So that's the graph representation. Then the neural developmental program, or NDP abstraction of that, is where we group related cells that participate in common developmental events. So these categories like A, ABA, ABP, those are all part of the same hypernode. And they're part of the same hypernode not because we felt like doing that, but because they participate in some common set of developmental events. So in this case, they form the AB sublineage in the lineage tree. Or so all the descendants of AB could be in a hypernode. All the descendants of ABA could be in a hypernode. We can organize the hypernodes in an arbitrary way, but they all have some common feature that they share. So they could be in the same sublineage. They could have the same function. They could all become muscle cells at their terminal differentiation stage, and so forth. So we use the hypernode methodology, then we use this neural developmental program to group cells into sort of their natural container, which is some developmental event. Sometimes that developmental event is obvious, and sometimes it's not so obvious. So we could have properties of a cell that, maybe like a genetic profile of each cell, a gene expression profile, where we can find like functional groups that we wouldn't be able to find, say, just by using the lineage identifier or thinking about what it might become in adulthood. This is a UMAP dimensionality reduction. So again, this is where we're taking this high dimensional data and we're plotting it in this two-dimensional space. So we have 5 variables reduced to a space of 2. So the 5 variables are up above. It's x, y, and z. Those are the sort of the volumetric parameters of position. Then we have a time parameter and a spatial transform parameter. So we have our 5 variables there. It's reduced to a space of 2 variables, x and y. And this is the UMAP reduction. So, it looks kind of like we have an embryo with clusters, but that's actually not quite what that's supposed to be, just given the methodology of UMAP. We use UMAP visualizations oftentimes to reduce things from very high dimensionality down to low dimensionality, and then try to find data points that are close together. And they're not necessarily clusters, but there is some similarity there. So we usually have some sort of relationship between the cells based on this UMAP visualization, where we can say this is a group of cells that are similar versus this group of cells that are similar, and what are the differences here. So, again, are there significant differences here between cells, or are there significant similarities? We can use this dimensionality reduction to find that out. This is another visualization of the hypergraph methodology. So, of course, this is worm development, where we go from a single cell to an adult worm. We then go down to this hypergraph neural network. And then, of course, you're using this attention mechanism with a projection head. And you're using that to discover these hypernodes where you have nodes contained within them. So one way that Label visualizes the hypernodes is by using sets intersecting or nested sets, semi-nested sets, to find nodes that belong, say, to a common hypernode, or sometimes nodes that are shared by more than one hypernode. And so he shows this diagram in this way. Usually if there's overlap, that means that there's a hyper edge that connects that cell is sort of in its position to connect two hypernodes. And so he can lay this out in a map, and it's kind of hard to visualize hypergraphs. So hypergraphs, of course, have these constituent nodes, and then of course they have hyper edges, which have they can have a lot of connectivity within each hypernode and between each hypernode. So that's really hard to visualize when you get above 5 or 6 cells, hypernodes. And so what you have there is you have this problem of visualization. And so this is his way of visualizing this, using these overlapping sets and then showing what nodes are within each hypernode and what nodes connect different hypernodes together. And you can see over time that this connectivity changes, that you get a more complex connectivity over time as you move towards the adult worm. This is moving to Jayadratha's work on DevoTG. This is the larval synaptic connectome that he's going to be looking at. So this is data from Whitfleet et al. 2021. They published a paper with larval connectome data, but larval connectome data at the level of synapses. So they were interested in looking at synapses and how those get remodeled over larval development. So if you're not familiar with C. elegans, we have a hatching egg. When that egg hatches, the worm enters the L1 stage of larval development, and then it goes through 4 stages: L1, L2, L3, and L4. Now there are forms of plasticity within that, so if you starve a worm in L1 stage, it will arrest its development, sometimes indefinitely or pretty far along. You can arrest worm phenotypes for up to 2 to 3 weeks using a starvation protocol. You can also, a worm can enter what they call Dower phenotype, which is a set of adaptations. It's what we might call diapause, and it goes from L2 to L4. So the worms have a different phenotype when they're in this Dower stage that is different from L2 to L4 development. In any case, this group, Whitfleet et al., they characterized the synapses. They looked at the changes in synapses across the connectome. Since we know the connections between neurons, we know where the synapses are, and you can just simply count the synapses and look at their turnover. And so these data exist, and so Jayadratha used this for his graph model. These are some graphs that he produced, sort of characterizing the data, validating the data, and then doing some other types of analysis. This is his visualization of the networks. So these networks are growing by over time. So you're observing over, various stages of development and into adulthood, and then the amount of time that's elapsed since the beginning. So if we start this video over, we start with birth at 0 hours, L1 at 5 hours, L1 at 8 hours, L1 at 16 hours, L2 at 23 hours, L3 at 27 hours, the adult at 45 hours. And you can see that there's a change in the structure of the graph over that time. Remember, we're representing synapses here. So synaptic connections of the C. elegans connectome. So you see that there is this, actually birth that really don't have a connectome, but that's sort of the baseline. And then we have this change in the topology. It's not immediately obvious until you get to L3 and then into adulthood. So you see some changes later, especially later in development. And that matches what we know about what's going on with C. elegans. There's still some morphogenesis going on in early larval development. And we have these stages of plasticity that occur, which aren't represented in this data set necessarily. But we do know that there is a lot of remodeling going on in the phenotype. And so as you get closer to the adult state, life history stage, you get maybe a denser network with more persistent synapses. This is another analysis he did. So he wanted to look at connection stability and look at like what's going on in L1 and L2 versus L3 in adult. So as you saw from the graph visualization, there's more sort of, it's a denser network in the L3 in adult. But if you use this AUC analysis, you get a slightly lower AUC value for L1 and L2. And this is interpreted as these more complex rewiring functions going on. So if you look at the network and sort of how it's changing its connectivity, there are more complex rewiring things going on in L1 and L2, so there's more turnover of synapses. There's more variation in synapse strength during that period than during the L3 to adult period. We also can look at some of the properties of the network connectivity itself. So there's this paper from Tolson et al. In 2013, where they proposed this rich club analysis of the C. elegans connectome. And basically it operates from the idea that there are these hubs in the network called rich club. They call it a rich club. There are these neurons that are that serve as connectors for the entire connectome. And so they are connected to a lot of other cells. And so we can take those neurons and we can look at those given this data set, and we can make some statements about, if we know that those hubs are going to be part of this, or those hubs are going to be part of this rich club, what is going on in development, especially with respect to synaptic connectivity. And indeed, we see some classes of behavior here. We have AVA, AVB, and AVE. These are all neurons in the rich club in adulthood. These maintain a high degree centrality throughout development. So they establish themselves as hubs early, and they maintain the centrality across development. Cells like RIM and AIB, those are also part of this rich club in adulthood. And over larval development, they gradually increase their connectivity. So in synaptic terms, they're not, it's not from hatch that they're very highly connected or that they're connected to a lot of other cells. This sort of increases over developmental time. And then you have fluctuating hubs. So you have hubs that are in the rich club analysis, but they fluctuate in terms of their connectivity, and they serve these transient coordinating functions in larval development. So sometimes they're more deeply involved in connecting things, sometimes not. So he was able to extract all these from the developmental synaptic connectome and then make this a comparison to the rich club. So that's our GSOC activity, and thanks to Jayadratha and Lewith for their work. We're working on papers, especially Jayadratha and myself. We're working on a paper for his work, his methodology, and some of his findings, and that will be out soon enough. One of the things we've been doing, though, this year as well is preparing AI summaries of our meetings. So, as you may be aware, our DevoWorm meeting occurs every week, and we've been doing this for over 10 years, but we've really been recording these meetings for about, putting them on YouTube for about 5 years. We started at the beginning of 2020, and we've gone to the end of 2025. So we have a lot of content on YouTube that needs to be analyzed. Quite frankly, it's very overwhelming to do it with a human because you have to go through all the topics and try to find the themes and kind of state what those themes are. So it's a lot of work. And so what we've tried to do is use Notebook LM to train it on the meetings, and we don't want to train it on all of the meetings from 2020 to 2025 because I think that would be a little not as useful. And so we've done this in 6-month increments. So we've been kind of working backwards from the latest meetings to the oldest meetings, and for a 6-month interval we'll train it on those meetings, and we'll produce one of these audio. They have this theme of like an audio podcast where they have two hosts or AI, AI personalities, and they discuss the content and they kind of give a summary, they give a title, they go through what was going on in the meetings. I mean, you can criticize how specific or vacuous it is. I mean, podcasts are kind of vacuous to begin with, but this is something that I think is a little bit helpful in kind of thinking about what we're doing at certain periods of time in these meetings. What are the overarching themes, and what can we learn from that? And it's hard to do with a human because with a human you can just make a list and you can kind of see the common themes, but it's not immediately obvious. You're going to get that sort of synthesis. You get synthesis, good theoretical synthesis, good topical synthesis from a human. It's just that it's going to be a lot harder to really, hit upon insights quickly. And so that's what we've done here. We have this one that's the Brainless Architects, another in DevoWorm Code of Development. And I've just taken some examples from the things that have been generated. And, just kind of interesting how it kind of names the episode and then goes through all the things that have been done. So that's something we've been experimenting with. I think it's been somewhat useful. We've been putting these on our YouTube channel, so you can go there and listen and see what you think, and then go back to those series of meetings. And if you find something interesting, go through, and there are descriptions on each meeting, so you can go find it fairly easily. We've also done a number of lectures over the past year. Actually, I'm counting from about November of 2024 till now, so that's been about a year. So in December of 2024, I did 2 problem-solving lectures, and this was like over the holidays. So one was on game theoretic development, and the other was on stress versus strain in a push-pull dynamical system. These are like lectures I gave on my laptop. I used the whiteboard, I drew out some diagrams, I reviewed some papers. The first one is on the work we've been doing on game theory and development. We have a technical paper on that and a couple other papers where we talk a little bit about how you can apply game theory to development. And those are on our publications page. Also, kind of working out some of these problems on a whiteboard. So you see a lot of cryptic things on this whiteboard example, but I promise you they make sense if you watch the video. This other one, stress versus strain in a push-pull dynamical system, kind of going through the biophysics of cell migration and of cell populations, and thinking about it as a dynamical system. So if we look at push versus pull dynamics in cell populations, whether you're pulling a bunch of cells apart or pushing them together, squishing them together, they give you different behaviors. You have different types of alignments, a lot of soft active materials, biophysics stuff here, and kind of thinking about how we can characterize that as a dynamical system. The reason I did this was because this year, in 2025, we've been working towards tensegrity models and figuring out how to specify those for development. Then on the left, I gave a talk, I think it was in April or May of 2025. This was on interactive morphogenesis. So this was on a topic called Moyer pattern formation. So Moyer patterns are where you have, say, like a set of concentric circles that overlap. And you can get like sets of concentric circles that overlap. And you can see this example here where you get this overlap and you get these patterns within the overlap that you don't see with either of the concentric circles. So it's very much an emergent property, a visual emergent property of those concentric circles overlapping. And you can do this for 2 or 3 or 4, so you can get a lot of interesting patterns just from the static case. But if you have a set of concentric circles that move against one another, so if you have 2 centroids that are moving towards one another or parallel to one another, then those concentric circles interact in very interesting ways. It's a dynamic model, and you can see that there's not only one pattern that emerges, but multiple patterns of motion. But the whole point of this, in talking about this in the context of DevoWorm, is that this is sort of this interactive morphogenesis. So you get this Moyer pattern formation, and the pattern formation is visual, but it's also, if you think about like some sort of process that's a depositional process that deposits things in concentric rings, or things that processes that overlap, then you get this opportunity for these emergent properties, for these interactive properties to emerge, and you get pattern formation in that way. And so I go through all of this in the talk. I also kind of compare it with reaction diffusion morphogenesis, which is where we have pattern formation, where we might be able to form stripes on a surface, where you have gradients that overlap, but the overlap defines sort of a boundary. In this case, we don't really have a strong boundary. The interaction, the space, or the boundary space are these interactions between patterns, and you get richer patterns out as a result. So I talk about some biological examples of this. I talk about how this might be useful in bioengineering and in other areas. And I talk about the mathematical structure. So it's very interesting. And we're working on this. I've been working on this area for quite a while. Don't really have a good paper right now, but that's probably coming soon. So yes, this year we did talk about tensegrity, and this is Susan Crawford Young's area of interest. I'm also interested in it, and Dick Gordon is also interested in it. And so we've been talking a lot about tensegrity. Mainly, how do you build models of tensegrity in development? So tensegrity is this idea of superstability. It's usually the way in which things are arranged. You usually have these sort of struts and connections between the struts. So you have basically this network where the forces balance each other out. So you have this dynamic tension that occurs in a tensegrity structure. And so there are a lot of different types of tensegrity structures that we can build. One, of course, is the geodesic sphere, which you see here. You see a number of geodesic spheres interacting here, sort of aligned. And those are all sort of tensegrity structures. So those are super stable structures, super stable spheres. And the idea here is that, maybe a cell membrane is a tensegrity structure. Maybe there are certain properties of cell membranes, maybe there are certain properties of different types of biological aggregates or tissues that have this super stability. If you put them under a fairly heavy load, they remain they respond with integrity, and that's an interesting thing for not only for regeneration, but for cases in which cellular systems or tissues are put under very high loads of stress or strain. And so we can use multiphysics to look at that, because multiphysics is where you have a bunch of aspects of physics that kind of come together. So multiphysics models are good at, say, modeling things like structural, thermal, flow, and electromagnetism properties of a thing. And so you can, this is different than a typical physics model where you're just modeling a small number of physical parameters and you don't have all of these things interacting together in the model. And so with cells, usually we're interested in structural and flow. We have some electromagnetism, and then we also maybe have thermal limits for tissues. But we have other things too that aren't in this diagram. So we definitely want to use some sort of multiphysics model. We want to be able to build these tensegrity structures. And we want to say that these things, which are usually things you find in the built environment, and also sometimes in chemistry, like buckyballs are a good example of where you find this in chemistry. But we want to say that we find this in biological systems, and we want to be able to model biological systems with tensegrity. Now the problem is that we can't model tensegrity using a typical multiphysics simulation software. And this has come into sort of the consciousness of the group by Susan. And she talked about how when you add multiple sources of compression to a structure, if you build a structure, because a lot of your multiphysics software, you can build a structure and it's like maybe like a building where you have the typical rules of a building that doesn't behave like a biological structure where you have like soft materials interacting and migration and things like that. But if you add multiple sources of compression to a multiphysics model, you end up with an indeterminate structure and a failure of the simulation. So we can't necessarily model tensegrities with multiphysics software or the typical multiphysics software. And so this is a problem one of the many problems we've been trying to overcome. But a word about multiphysics, why don't we just take another route? Why don't we just use another method? And the answer is that multiphysics are necessary to model ultrastability in embryogenesis. So if we're thinking about things like liquid flows or flows of cells in a liquid, or rearing temperature, or building upon existing structures, or bioelectric dynamics, we want to be able to use multiphysics models to model all of those things in their totality, because they're all important and they all play a role in this these ultra-stable structures. So another thing we're doing with these tensegrity models is we're modeling vertex biology. So vertex biology has become an interesting niche area in the last several years of cell biology, where different groups have identified, especially when you're looking at tissues in development or tissues, cells in a tissue, you have these different ways in which they're oriented to one another. And one of the things that plays a big role in their stability is the existence of these junctions. You have examples such as bijunctions or trijunctions. If you see these models of cells, they adhere to one another at these junctions. And sometimes you have two cells that adhere together. Sometimes you have multiple cells that adhere together. And they form these junctions that actually have some interesting geometric properties. So a trijunction forms this sort of stable, instead of like a bijunction or some other type of more transient structure. And so that's why these kind of junction systems, these end junction systems, are interesting. Bi and trijunctions of cell membranes provide points of ultrastability and cell sheet dynamics. And this is maybe a route to biotensegrity that doesn't necessarily get around the multiphysics models, but it helps us understand the context of the multiphysics models more clearly. And then, of course, you have these structures of cells with these different junction types, and they're subject to flow. They're liquid flow. They're subject to shear forces from the liquid flow, and you have shear forces that act as sort of a stress or as a strain, and then you have the stress and strain at the bi and trijunctions. So you have these stress strain curves that you can derive from these different physical attributes of a cellular environment, multi-cell environment, and we can say things about that tissue based on that map. These are tensile and compressive elements, so our bars here and our connections between the bars, these look like networks. If you recall from when we were talking about hypergraphs, we have nodes at the end of these bars, and we have these compressive elements that are, so we have the tensile elements and the compressive elements. The compressive elements are the bars, we have the nodes, and then the tensile elements are the connections between the nodes in green and in red. So we have these different structures. They can become quite topologically diverse. They also can twist and bend and maintain their stability. And so Susan's identified two structures from her thesis that are suitable to this. We have the T3 type, which are these 3-bar structures. You see these up here on the upper right, where you have the black bars, which are these struts, and they have a 30-degree twist, so they're twisted almost like a helix. And then you have these tensile elements connecting their ends. And so that's supposed to form an ultra-stable structure. We also have these T4 structures, which are 4-bar structures. So the blue bars here are oriented at a 45-degree twist. And then we have these tensile elements, the green and the red, that keep those stable. Ultra-stable, in fact. We can have symmetrical transformations along 3 spatial dimensions, so we can twist them and keep twisting them back and forth. And this represents sort of the dynamics of a tissue where things are moving around, and we don't want to just assume that they're static. We have this dynamic model of tissue and of organs that move, and of course cells have to maintain their connections to one another, the junctions between them, and everything is kind of moving in that way. So we need this ultra stability to be biologically plausible. We have these transformations along 3 spatial dimensions. And as you might imagine, because we can model this in time and there's all this constant movement, it provides a large amount of configurational complexity. Some of that is viable and some of that is not viable. And this is the problem. This is almost like protein folding in the sense that it gives us this large configurational space that we have to sort through to find the the most stable path through this conformational space, the most viable path. And so, like protein folding, you can model these things twisting, and you need to model all the conformational states, and it's a hard computational problem. So this is another thing we're running up against, is this configurational complexity. This is an example of scutoid epithelial cells. So you saw some of these hexagonal structures in the last image. That's sort of one of these scutoid epithelial cells. That's kind of our model for all of this work. It's not just the top of the cell, which is the hexagon. You also have this 3rd dimension, which goes down. And so you can see that you have a series of trijunctions here, and they're connected not just at the top, along the top of the sheet, but they go down quite a ways. And sometimes those connections down below are in a different location than the trijunctions on the top of the sheet. So as they go down to the substrate, they twist and bend. And so this is what we're trying to understand, is how these cells bend as they're sort of maintaining contact. So these scutoid epithelial cells exhibit helical twisting in a sheet during migration or growth. And then these sheets, of course, exhibit trijunctions that bend and twist from the top of the sheet to the substrate. And this actually gives us 5-dimensional dynamics instead of 3, because we have the 4th dimension of time, we have changes in the shape, and then we have this twisting, which is a 5th dimension. So we have our volume, or XYZ, that's position, and then we have time, so we can count our temporal period, and then we have division angle, or contact angle, which is how much these things twist over time. So we treat time as one dimension and that twist as another. The angle, or the change in the angle, I should say. And so this is from this paper in Current Biology that talks about dynamic changes in epithelial cell packing during tissue morphogenesis. And they kind of go over sort of the complexity of this problem. We've also done a lot of work in terms of looking at C. elegans syncytium. So C. elegans has a number of syncytium in the biology of the adult C. elegans. What a syncytium is it's a group of cells that, of course, we know from the lineage tree that cells divide in development. But at some point, for some structures, the cells fuse back together and form what they call these syncytium, or single cell bodies with multiple nuclei. And so this is something we're interested in. There are a number of famous syncytium and C. elegans. We'll talk about a few of those. But the idea is that you have this single cell body with a bunch of nuclei in it. And that results from cell fusion. But sometimes the nuclei can divide independently of cell division. So if you fuse the cells together, you can still get a doubling time of nuclei within that structure. And so this is something we wanted to understand and think about it in terms of lineage tree representation, and then maybe what's going on in terms of the cellular dynamics. So what I just showed you a picture of is the rachis. The rachis is sort of a bridge for oocyte growth. The rachis comes together from germ cells that fuse together, and you find this in the towards the distal end of the adult worm. You have these rachis and rachis bridges, and they have, as you see, you have these multiple nuclei within a single cell membrane. And so the rate of cell fusion and nuclear division governs formation of the syncytium, and we could use a regular lineage tree for that, but as we'll see, it kind of violates the convention of the lineage tree. So there are ways to get around that using reticulating trees. This is the origins of the vulval syncytium. This kind of is put together from data from a series of papers where we have the developmental cells in the C sublineage. So we have a couple of division events where we get cells that are in different, several division events where they have different division orientations. And then eventually in development, different stages of development, a lot of them are actually in early development, within the first 250 minutes, form these hypodermal cells. So in this case, we have the C sublineage, which divides. So we have 4 cells that end up fusing together into a series of hypodermal cell bodies. And this is this is CAA, CAAP, and then another part of the C sublineage, we have CPAA, CPAP, and they form these hypodermal cell bodies at these different points in development. So that's this diagram above. So we have to have this reticulating tree where cells divide and then they fuse back together. Then we have, to form the vulval syncytium, you have fusion events at different points in development. So you start off in the 220 minute to 235 minute range, you get a bunch of these cells established. Then in the larvae in L1 and L2 and L3, you have different syncytium that form these hypoderm 7 cells that form from different parts of the lineage tree much later in larval development. And then finally in the adult, you end up with the vulval syncytium as an organized structure around 2,780 minutes of development. So that's actually an late L4. And so we can draw out this lineage tree by going back and looking at how we where we start with, we get these division events, and then we get these fusion events. And it's not enough for the cells to fuse together. They actually have to form a structure. So you see that the structure is formed here from these fusion events. And those fusion events form the shape. So they form these different rings of the vulval syncytium, and they get laid down in L4. And then by adulthood, you have this structure. So this is kind of a reticulating lineage tree coming down to the structure. Again, this is not like our final product. We're just kind of thinking about how to do this, how to represent these things using the lineage tree, but modifying it to account for these syntition. So thanks to our contributors for 2024 and 2025. And again, our meetings are on YouTube. We have weekly meetings. We have other content on YouTube. And so definitely check that out. We covered a number of topics this year, including Lagrangian and Eulerian models, biotensegrity, multiphysics, peptide modeling, the limits of simulation, something called swarmolators, developmental graphs, CompuCell 3D, which is a modeling platform, soft materials, transcriptional budgeting, node layer duality, vertex biology, discrete and continuous hypergraphs, cybernetic flocks, evolution beating artificial neural networks, non-random embryo networks, and macromolecular crowding, among many other topics. So our meetings are usually where we talk about our updates, and then we get into some set of topics. Usually it's a paper that we review, but sometimes people give presentations on different topics. So if you're interested in giving a presentation to our group on something you're interested in, you can see kind of what the trend is here. It's sort of biophysics. It's different types of mergers of physics and developmental biology and cell biology, organismal biology, processes like development and evolution, and of course complexity, where we talk about emergence and other things like that. Please contact us if you're interested in presenting, and thank you for a great year, and I hope to see you next year.